In this module, we're going to be looking at performing differential expression across multiple datasets. In order to do this, we again need to select across datasets, and now we're going to select the MegaSearch function, which allows us to find differential expression between datasets. And we're going to click Next. And here again, you're allowed to select what platform or type of data you're going to look at, and we're going to look at the datasets that are run on the Affymetrix U133P2 platform. Click Next. And again, now you're going to be able to select which datasets you would like to compare or look at. So just as in the MegaSampler module, we're going to select the same datasets. So we're going to select a tumor dataset consisting of 204 breast tumors, and also a colon dataset consisting of 290 colon cancers. And then we will also make comparisons with the neuroblastoma 88 tumor dataset that we've used multiple times before. So we're going to select these and click Next. So now you can see that we are presented with the three datasets we've selected. We'll leave the transformation to log2 of the expression. And now we want to compare breast and colon tumors to neuroblastoma, so we just need to select which groups to put these datasets in. So we'll leave breast and colon as in group 1, and we will select neuroblastoma as in group 2. You do also have options to put on filters. For instance, if you were only to want to look at how breast and colon tumors compare to an amplified or non-amplified. So there are options to use tracks to select filters. However, for this example, we're not going to do this. So we're going to select neuroblastoma in group 2 and submit. And here you can see that they have now divided the datasets into the respective groups. So breast and colon are both identified as group 1, neuroblastoma is group 2. You do have options to select a subset of genes if you only want to look at a specific gene set and how it is differentially expressed across the datasets. And another aspect that I can point out here is you do have the Hugo Once dataset. And what this does is it selects which dataset will define what probes are used in the analyses. And because we are going to be doing comparisons with specifically neuroblastoma, we're interested in neuroblastoma for this analysis, we will select the neuroblastoma dataset to define which probes are used. And we will then click Next. So this analysis does take a little bit of time, considering that you're looking at all genes across multiple datasets, so across multiple samples. So you just need to be a little bit patient for the results to come up. So now that your data has been generated, you can see that you had a total of 582 samples, with the 498 in group 1, which is shown up here. That consists of the breast tumors and colon tumors, and you have the 88 tumors of the neuroblastoma in group 2. And here you can see that then all the gene expression is split into those that are highest in group 1 and those that are highest in group 2. And for those who are familiar with neuroblastoma, you can see that there are quite a number of common neuroblastoma marker genes or genes involved in neuroblastoma development, such as FOX2B, DDH, FOX2A, falling out in this list, stating that they are higher in neuroblastoma than in breast and colon cancers. You can also see on the right side of each of these tables that you're presented with the average expression in group 1 compared to the average expression in group 2, rounded to the nearest whole digit, so that you get an idea of the difference in expression. So for instance, for FOX2B, you can see that the average expression on a log2 scale is 12 in neuroblastoma compared to 3 in the other tumor types. Again, you can then click on the specific probe, and this will generate a similar cross dataset plot that we've seen before with associated statistics, and we can then scroll down and adjust the plot as we wish to view it. So again, we can put neuroblastoma into the first position, and then we can put breast in the second and colon in the third, and we can change the colors again as we see fit. So we will just be able to visualize it better and redraw this graph. And here you now see that we've put neuroblastoma with the highest expression of FOX2B in the first column compared to breast and colon in the other one. So this is just another example of how you can use these cross dataset analyses to look at gene expression. However, we must again point out that we have not done any normalization across the data sets, meaning that we assume that if the data sets are run on the same platform with the same data handling, that we then can make preliminary comparisons across data sets. However, this must be pointed out that this is not the optimal approach for looking at gene expression between individual samples or getting definitive answers, as there can be discrepancies between how the data has been produced.